Hi friend, this is Vaibhav Konduskar. Welcome to our YouTube channel Pharma Knowledge. Today's topic is the guideline on WHO guideline on the general guidance on whole time study, that is WHO TRS 992 Annex-4. So before starting this video, if you are not subscribed my channel, please subscribe the channel and share the videos. सो ये टॉपिक स्टार्ट करने से पहले हम लोग पहले ये देखेंगे कि एक डे टू डे लाइफ का एग्जांपल। जब भी कोई चीज हम लोग मार्केट से खरीद लाते हैं, तभी हम लोग जनरली बोलते हैं कि इस चीज को हम लोग कितने दिन तक स्टोर कर सकते हैं। मतलब क्या है कि उस चीज का होल्ड टाइम, सपोज जो चीज है उसको 7 डेज तक हम लोग स्टोर कर सकते हैं, तो 7 डेज उसका होल्ड टाइम हो गया। मतलब उस 7 डेज में उस चीज को कुछ भी नहीं होना चाहिए, वो एज रहना चाहिए, इसलिए उसको हम लोग होल्ड टाइम बोल सकते हैं। तो यही सेम फंडा है फार्मा में। तो देखेंगे एक्चुअल होल्ड टाइम स्टडी क्या है और उसको कैसे परफॉर्म करते हैं। First of all, we will see the introduction of the whole time study. So manufacturers should ensure that the products that they manufacture are safe, effective, and of the quality required for their intended use. Means whatever the product are manufactured by the manufacturer, that product should be safe, effective, and quality product for their intended use. Then the good manufacturing practices require that arrangements should be made to ensure that the dispensed raw material, then packaging material, intermediate products, bulk and finished products are stored under appropriate conditions. Means raw material, packaging material or intermediate products or bulk finished products that should be stored under the appropriate environmental conditions. Then the choice of maximum holding period should be supported by the relevant data. Means it should require some study for the maximum holding of that particular material. Then next is the terminology related to whole time study. Means this bulk product and intermediate, this is very basic terminology and this is very important terminology related to the whole time study. What is the bulk product? Bulk product means any pharmaceutical product that has completed all processing stages up to but not including final packaging. Means it is the awaiting for the packaging. So that is called as the bulk product. Then intermediates. Intermediate means partly processed product that must undergo further manufacturing step before it becomes a bulk product. This is two important terminology related to the whole time study. Then next is the aspect to be considered for whole time study. These are the different aspects to be considered for the whole time study. First thing is the whole time can be considered as the established time period for which material, that is dispensed raw material, intermediates and bulk dosage form awaiting for final packaging may be held under specified condition and will remain within the defined specification. This is very much important aspect related to the whole time study. Means whole time means it is the established time period for any material. Means it may be a dispensed raw material or intermediate products or bulk dosage form awaiting for the final packaging. It's held under specified condition, means it is held under the specified condition. But whatever time period you have held for that particular material, in that period that material should not be changed any defined specification. This is very much important sentence or important aspects of the whole time study. Means then next is the whole time should normally be determined prior to marketing of the product. Means before marketing of the product you should define the whole time. Then the whole time studies may be included during development on pilot scale up batches or during scale up, and it should be confirmed during process validation of commercial scale processing. Means these are the different aspects of the whole time study. Here I given the example. This example is given in the WHO guideline. Different stages of the whole time study and which stages to be considered for the whole time study, I have explained in this slide. First is the binder preparation to granulation, just consider as the granulate. In the wet granulation to dry, consider the dried granulates. In the dried granules to lubrication slash blending, consider the lubricated blend. Then the blend to compression. Then the compression to coating, consider the core tablets. Then the coating solution to preparation, consider as a coating solution. Then the coating to packing, consider the bulk coated tablets. Then the coating to packing in bulk. Then the packing of bulk to finish pack dosage form. These are the different stages to be considered for the whole time study. Here I explained regarding the oral solid dosage form, that is the tablet. So whole time you can define, means every dosage form need to define the whole time period or any stage of the dosage form. So here I explained regarding the tablet dosage form. Then next is the how to perform the whole time study. So for whole time study, to perform the whole time study, it requires the written protocol, then the procedure or program should be followed. Means it requires the protocol. Then the what are the content of the protocol, whole time protocol? First is the title, then the reference number, version, then date, objective, scope, responsibility, then procedure, description of material or product, then sample quantities, sampling method and criteria, acceptance limit, frequency of sampling, sampling locations, pooling of samples, storage conditions, type of container, method of analysis, results, conclusion, recommendation, signatures and dates. All these basic content required in the whole time study protocol. So these are the very basic points, bullet points to be considered in the whole time study protocol. Means what are the objective of the study, why we are performing this study, then the scope, what are the responsibilities of the team, then the which materials we have selected for the study or product, then the what is the sample quantity, then the sampling method, then the acceptance criteria for the testing, then the method of analysis, then the frequency of sampling, then the storage condition. All these things are required in the whole time study protocol. Then next is the acceptance criteria are typically more stringent than register specification. Why this is important? This acceptance criteria is required more stringent because it gives the more assurance or more confidence that the material is well within the control. Then typically one or more batches of a material, intermediate or product can be used for the determining the whole time study. Then the hold period for each category of material should be established on the basis of the study by keeping the material in either the original or simulated container used in the production. This is very much important clause for the whole time study. Means simulated condition or same type of container required during the whole time, which we have using during the actual production. Then the headspace of container used for bulk storage in manufacturing and or quarantine is important. Then next is the what are the whole time stages, then which test to be carried out, and what is the study time for the whole time study. that all these things are explained in this slide, and this chart is given in the WHO guideline. First stage is the binder preparation. Which test to be carried out as per specification? The microbial test, appearance, viscosity if applicable. And study time is the initial, then 2 hours, 5 hours and 8 hours. If your formulation contains the starch, then the study time is the initial 2 hours and 5 hours. Then second stage is the dispersion prepared, including granulation paste, coating solution and coating suspension. Test to be carried out as physical appearance, specific gravity, viscosity, sedimentation, pH and microbial test. And study time is the initial, then 12 hours, 24 hours, 36 hours, 48 hours, 60 hours and 72 hours. Then third is the granules. Test to be carried out as description, assay, RS, LOD, water content, particle size distribution, and physical properties like bulk density, tap density, and angle of repose. The study time is the initial 15 days, 30 days, and 45 days. Same for blend. Test carried out to be carried out as microbial test, LOD, blend uniformity, particle size, then bulk density slash tap density. And study time is same, initial 15 days, 30 days and 45 days. Core tablet, next stage is the core tablet, uncoated in bulk container. Test to be carried out as description, hardness, thickness, friability, DT, DISO, then the assay, degradation, RS, then the uniformity of dosage unit and microbial test. And study time is the initial 30 days, 45 days, 60 days and 90 days. Same for coated tablets. Next stage is the coated tablet. Test, description, appearance or visual examination, hardness, thickness, friability, DT, DISO, then the assay, degradation, then RS, moisture content and microbial test. And study time is the initial 30 days, 45 days, 60 days and 90 days. These are the study time for the whole time. All these things is explained in the WHO guideline. So also in my book, Pharmaceutical Quality Assurance book, I have explained this topic in very well. So you can refer this book also. This book is available on the pragationline.com website. So you can refer this book also. So thank you. Thank you very much.